Here we go. This is the Advanced Science Academy Journal Club number 19. Brandon Billack is going to be presenting on some of the new research on ketamine, which is widely known as a horse tranquilizer. It's used in veterinary medicine quite frequently and has been for a while. It's also known on the streets as Special K. And those of you who live on the streets, you know just as well as I do what the drug names are. Okay, Brendan, it's all you.
Sharing it. So the paper is on effects of early ketamine exposure on cerebral gray matter volume and functional connectivity. So this is just a quick background on some the brain and how ketamine and A also has LSD and DMT, how it affects the neurotransmitters. So ketamine targets the NMDAR and AMPAR receptors, and it releases glutamate.
I think that's the other way around. They're NMDA receptors. That's what the R stands for.
Oh,
And they receive glutamate.
it says glutamate release.
Yes, from the previous neuron.
Oh, gotcha.
Increases in glutamate release will affect the NMDARs, and those, see, if you follow the arrows, those affect the increase in brain-derived neurotrophic factor, BDNF, which causes brain cell formation.
Mm-hmm. So ketamine and other hallucinogenic drugs. Just read through this here. Release glutamate, and it leads to an activation of AMPA and NMDA, which is what it shows in the figure. A and B. B just shows the subcortical areas instead of the brainstem. So, as ketamine blocks the NMDA receptors on the cortical pyramidal neurons, the increased glutamate release in the cortex stimulates cortical AMPA more than NMDA receptors. And a little bit more background on it. It's a Schedule 3 drug, and it's known for its dissociative properties and hallucinogenic properties. So this is pretty much the main information, the main point of the study, which is the regional gray matter volume differences between ketamine users and non-ketamine users. And then there's also, they made a differentiation between people who started using it in adolescence and people who started using it as adults. And these values basically show, they're going to be shown in the figures later, but basically they demonstrate the differences in functional connectivity of these two parts of the brain down here, and also the regional gray matter volume differences between users and non-users. And from the table, basically, users have less regional gray matter than non-users, and we'll get to that in the figures, exactly which parts of the brain. In regards to the connectivity, users seem to have a bit more. That's going to be in the figures down below as well. And the statistics and data were corrected for multiple comparisons using the Monte Carlo simulation. And what they corrected it with is right there down below, P alpha. So figure 1, got Marco on that.
So essentially what we see here is the ketamine users have lower gray matter than those that did not use ketamine in this study. And yeah, I think that's the entire figure.
And also we got up at the top with the pictures of the brain, the highlighted area is where the gray matter differences are. And on the right here is all the parts of the brain and what they do that are listed in the figure. And as we go down, I'm only going to include the new parts mentioned.
Wow. Let's actually go over those because that's kind of interesting. It's also kind of interesting which sides they chose. Left here, right in the next figure, right?
Yes. And this one also includes the right insula.
Yeah. So tell us what each of those does, and maybe we can add some discussion about experientially what it causes.
So it's organized a bit weird. The right insula is the first graph there, but it's the last figure. So besides that, they're in order. And the left inferior parietal lobe is involved in perception of emotions and interpretation of sensory information. The left dorsolateral prefrontal cortex is associated with executive functions, including working memory and selective attention. The left medial orbitofrontal cortex is a key brain area on emotion and the representation of reward value. And the right insula is engaged in interoceptive awareness of emotions such as thirst, pain, and fatigue, and the ability to time your heartbeat.
So if we think about the effects of the drug ketamine, one of its key features is it causes dissociative effects. Do you guys know what that means? Dissociative?
Like where you don't feel connected to your body.
Yeah. Yeah,
you
like
feel
literally.
Multiple things at once.
Yeah, yes, that too. Yeah. Yeah. And so if you look at the brain regions that seem to be affected or activated by this or reduced, they kind of have something to do with that. So like perception of emotions in both the inferior the parietal lobe is your sensory lobe. Like that's the thing that's really integrating senses of the outside world. But you add in the right insula, which is your own awareness of yourself. Your awareness of yourself is changed, including your pain. Actually, one of the reasons that it works so well on pain, ketamine, is that you might still feel the pain. You just don't realize that it's you. Like, yeah, there's pain there. Oh, is that me? Oh, so, let's see, what other interests? The DLPC, the dorsolateral prefrontal cortex, is probably the most studied area of the prefrontal cortex having to do with executive function, decision making, moving one's body around. It's studied in cats and monkeys and humans for what we call saccades, darting your eyes to one spot or another. Like if something shows up here in the screen, your eyes move to that, but you have to make a decision to do that. And that's the area of the brain that actually does that. Let's see. And then the orbitofrontal, which has come up a bunch of times in Journal Club, that's emotional centers, so limbic system. Can I just, well, you know what, I'll come back to why, I'll throw in my 2 cents about what I think about the quality of the figures later. So you guys can keep going.
All right, great. Figure 2 is Alex on. RC is doing figure 2.
So these are showing different like cross sections of the brain and the different users, and I believe the top is showing the actual grading of the connectivity. So in ketamine users, there's a increased functional connectivity. So it's like slightly better than the non-users, and then on the bottom it actually shows the activity. I don't, um I don't know if that's the right word or not, but it's like showing how in the ketamine users versus non-ketamine users, there's like an increased uh just like connectivity in like their brains, like from the use of the drug.
So anatomically, Brendan, if you want to go over those.
Yeah, so I didn't include like the right insula again or anything like that, just the new ones. So the right superior temporal gyrus contains the auditory cortex, which is responsible for auditory processing, and it also is involved in processing social cognition. And the right inferior frontal gyrus is associated with various cognitive functions, including attention, uh, motor inhibition, as well as social cognitive processes such as speech. And the right inferior temporal gyrus is responsible for visual object recognition, and it receives processed visual information.
Okay.
Moving on to figure 3. So Alex was going to read that. I don't know if he's on.
Yeah, so I believe the bottom graphs just show the volume, the regional volume, for those who have used ketamine and those who haven't, and then the control group. And then at the top it shows the left precuneus. I think that's just the volume of that in the different studies or the different groups.
The little like lit up area is like where it is.
Yeah.
Is where they're measuring. And adolescent users that started before they were adults have less gray matter in total than people that started when they were adults, and both those groups have less than the healthy people that did not use ketamine. And the left precuneus is involved in memory tasks regarding verbal responses about spatial details. And figure 4, I'm going to do that. The right precuneus is used in memory and spatial function, and the table here, again, the highlighted areas are like the orange areas is where they're measuring from, basically. And these two graphs demonstrate the connectivity between users and non-users, and this one's interesting. So people that started using when they were adolescents have greater connectivity than those in the control group, and so do adult users, but adult users have even more connectivity than the adolescent onset users, which I guess thinking about it a little more makes sense because the adolescents that used have less gray matter than the adults that use it. And that's for both of them. Both graphs right there.
So let me pipe up and can you go back to figure 1?
Yeah.
So for those of you who are doing lab reports, for me especially, I just want to go over some stylistic things. One, I don't really like.
Well, the graphs are in order.
That's really obnoxious. Yes, I agree. We were looking at that yesterday. I'm like, why can't they order them the same in the top as they do in the graph? I don't know. And that's a minor picky thing, but generally your y-axis should be there. It's labeled, but there's actually no axis there. And you've got what we call minor x-axes. That's at each decimal, 0.2, 0.4, 0.6, 0.8. And those are these minor axes that I think are really ugly and don't really help us that much, because all you have to do is show the asterisks that things are significantly different. And so freshmen, we haven't gotten to do this yet in class because of COVID. But those of you who had me previously know that we learn a little bit about statistics, and one of the ones that you learn about is an ANOVA, which is an analysis of variance. They ran something very related to that called an ANCOVA, which is an analysis of covariance. And then they did a post hoc test. So the analysis of variance is what we call an omnibus test. It's just saying, is there a difference or is there not? And so let's say it's significant. Yeah, there's a difference. It doesn't actually tell you in what direction it goes. Like what, okay, yeah, the statistics are saying there's a difference, but it's completely meaningless unless you're actually comparing them to each other, which is what the LSD is down at the bottom of the figure legend. LSD is a post hoc test. You guys, at least AP usually runs a Tukey or a Dunnett. LSD works especially when you have groups of 3. Here they ran it on a group of 2, which I find odd, but whatever. So each of the lit sections of the brain are coordinates. And that's actually also what you're seeing labeled above, like x minus 40, x minus, what does it say, 19? With a t-score, showing you kind of like what area it is and how powerful the effect is. If you go to the next one. Okay, figure 2. Same idea. Multiple. So in figure 2, I believe they're showing this is those brains you're seeing are actually not a single person's brain. They're actually an averaged brain across multiple groups and corrected for age and gender and stuff, because there actually are sex differences in anatomical sex differences in the brain that need to be corrected for in order to find correlated areas of the brain. So whenever you hear someone tell you there are no differences between a male and a female brain, they're idiots. There are significant differences in anatomy and the size of certain parts of the anatomy, and noticing those differences is not saying that one is better than the other, which actually kind of gets at one of the weird things I think Brennan's going to talk about when he gets to his conclusions. See, other things I don't like how they write over the bars. It's really hard to see. Don't do that, please. It's almost like that was coding used when they were designing it and then just never removed it, which is really ugly. Go to 3. Here's one that I think is really tacky. In figure 3, do you see there's a reflection under the XYZ? Why? Why is that? What, like, it's supposed to be artsy fartsy, or it's supposed to somehow, you do not want to distract away from the data. You want the data to be the highlight, not your artistic skills. Okay, so that's bad. But this was one of the more interesting results, right? If we're looking at regional volume and you're seeing, as was said, the in the left precuneus, you're seeing connectivity differences later in the next figure, as well as the regional volume. Adult onset has a larger volume than adolescent onset. Go to the next one. But then in figure 4, you see that the connectivity is also increased in that adult onset relative to the
adolescent.
Now, what I just said, you see an increase in activity in the adult versus the adolescent onset. Am I right?
Yeah.
Am I?
I mean, the control group has comparatively less connectivity.
Yeah, but if you look at where the comparisons that are being made, both of them are being compared to the control, but there is no significant difference between them. Even though you can see it looks like there's more.
Well, there's a tiny difference, but it's not
But
marked.
it's not a difference. So that's the thing that we have to. That's hard to, when you're in high school and even in college. It's hard to wrap your mind around this. If the statistics are saying that they are not significantly different, then they are not different. The difference that you see is probably then indistinguishable from chance. It doesn't mean it's not different in reality. It just means that you can't really prove it. So in this case, there really isn't a distinction between the two of them. And you actually see in the right figure, there also is, even though it looks like the orange bar is a little bigger, that they can't rule out that that's just due to a coin toss, like just chance. Okay. So those are just my 2 cents on the way the figures were designed. I also don't really like, this is really, really freaking picky. I don't like using serif fonts in figures. You guys know what a serif is? Getting no responses from anybody or their ceilings, because I'm mostly looking at ceilings. A serif is a little tick kind of on each letter, and instead of just a block letter, they're the things that make on typefaces that make them look a little fancier. So there's a big difference between where it says in Brendan's slide. Look at the two places it says right precuneus up at the top. So in the white letters, right precuneus in the figure, that is a serif font. It has all those little kind of extra shadowy parts, and they're called serifs. And then on the right, in his what he was doing, his list of anatomy, it says right precuneus, that in a sans serif font, one that does not have serifs. That's Calibri. And you may not think that that is important. However, there have been studies done on how much the human brain is able, how fast it's able to read, how much it actually likes, the emotional reaction that certain typefaces evoke. So when reading some sort of sign or figure like this, it's easier to read sans serif fonts. But if you're reading a paper, prose, text, it is actually easier for the human brain to read serif fonts, which is why they were developed. That was a big, big advance in computer science, by the way, because the original computers all had one typeface. There was no way to change it. It was a word processor, and that's it. But then Microsoft came up with a way to show these little serifs and stuff, and it changed typography. That is really going far afield, but I think that's kind of interesting, especially if you're into publishing. Juniors who are in this AMI now, those little differences are things you're going to really focus on next semester when you take the capstone course, the first part, which is the editing and publishing. Okay, go ahead, Brendan, finish up.
Sure. So there's two main implications here. And the first one is that due to the differences in gray matter between someone who has used ketamine and someone who hasn't, a young brain will definitely develop differently, like having taken ketamine than having not. So someone will develop differently, like they will turn out, I guess, somewhat differently if they take ketamine, like having not. But what
that's kind of what I was getting at, right, Brendan?
Yeah, what difference there isn't exactly known.
Yes.
Because it doesn't exactly, there aren't any noticeable effects because of the gray matter loss, like in particular, but there is gray matter loss, so there is a developmental difference. And the second one is that ketamine's therapeutic potential is going to be limited by any adverse effects it has on brain development, which again aren't exactly known quite yet, except for the gray matter being lost with use. But it's not quite known exactly what that does.
All right, if you want to unshare your screen, I just want to get people's perspective on this. Would it be fair of me to say that I talk about drugs more often in my classes than any teacher you guys have ever had? Is that fair? I think that if I were a parent and I was listening in to the way that I teach, I might find it both entertaining and maybe a little um, worrisome because it could at times sound like Charleglio is normalizing the use of drugs or normalizing what they do, and so I think it is incumbent on me to make sure that I make the point when things like this come up. I make the point that illicit drugs are bad. Okay, we know that they, like ketamine, can change your brain permanently. Ketamine's an interesting case because we're not really sure if it definitely makes changes developmentally. We don't know if those changes are necessarily a bad thing. In fact, Brendan, did you want to say something about like why they should care about ketamine, like its future and stuff?
Well, I think it's important because like it has potential as being a therapeutic drug. Like it can be used to treat pain and such like that, but the problem would be the effects on the brain. Like what Dr. Carr and I said already, like it does decrease the volume of gray matter in the brain, but it's not exactly, there haven't been studies done on what that actually does, like how negative those effects are.
Yeah. Okay. So I'm going to be really obnoxious, people, because I want you to talk. Design an experiment where you'd be able to actually answer that question. Design an experiment, okay, is the beginning of every advanced biology course exam you will take in college and beyond. Okay, everyone is like, here's your take home exam. It has 5 questions. You have all weekend to do it. And you're like, all weekend? Wow, for 5 questions. It's going to take you all weekend. They're really hard. And they're almost always design an experiment that. Okay, design an experiment. And you can do it right now. This question I think is easy. Design an experiment in which you're able to answer that question. What effect does having the drug in adolescence have behaviorally on a person relative to not? How would you design that experiment? Be crazy. Doesn't have to be feasible. Just come up with a crazy experiment. This is the fun of it. Anybody? How would you do this experiment? Okay. I'm going to pick on somebody. I'd like you guys to be on camera. You know about that. That's a respectful thing. AJ, give me something.
Could you maybe do an experiment about the effects like on the brain like of somebody or of like a brain that an adolescent brain that is not addicted to drugs and one that is addicted to drugs?
Sure, you could do it independently where you get an idea of, oh, well, this group of people versus this other group of people. But I want to really know what effect it's going to have on your brain. What you just described. Okay, so AJ's a freshman, and again, I haven't gotten with the freshman. We haven't done any statistics or any of that lab stuff yet because COVID. But for those of you who have had AP Bio with me previously or some other course, Honors Bio, remember the difference between independent samples and matched samples. He just gave an example of independent samples, which is a totally valid way to do this. Take a group, take a different group, give one drugs, don't give the other group drugs. The problem is that you're comparing kind of apples and oranges, right? I wanna know about apples to apples comparisons. Is there a way to do it?
Are there ethics involved, or like we ignoring?
No,
ethics?
I don't care about ethics. No, you can be a
Two
Nazi.
identical
Well, that
twins.
changes a lot, actually.
Hold on, I didn't hear anybody. Wait, what'd you say, RC?
You take two identical twins and addict one of them to drugs.
Exactly.
Yeah, without ethics involved, like it's actually possible to do a study like this.
Yeah. You could also, without the laws of physics, you can also just go back in time and do the same person again, right? But laws of physics being constrained here, you could take twins, right? Which is what I talk about all the time, right? I don't like my sister Katie. Sarah, I can deal with, right? But all through Katie's childhood, I beat the crap out of her, and so now I know, well, what happens to a child and adolescent brain when it's constantly abused, right? Not an ethical experiment, but one I could have done because I have identical twins. But they do have to be identical, right, RC?
Preferably.
Yeah, preferably. I think really kind of it has to be. And twin studies are really, really important to understand behavioral differences, because identical twins are remarkably similar in their behavior. And we know this because of identical twins separated at birth, which doesn't happen much anymore. But in the early part of the 20th century, that happened all the time. Two babies were taken from a mother who was considered unfit and were separated, and then those babies found each other or were studied by neuroscientists. They weren't called neuroscientists at the time, so they would be called, I don't know, psychosociologists or whatever, and they would study how amazingly similar the two were. So if you were to have one brain develop on ketamine versus the other one, you might get an idea of what that difference in ketamine developed brains is behaviorally. But that's the weird thing about this study. Like, okay, the question I have to ask everybody, and Brendan especially. Brendan, should this have been published in Scientific Reports? Scientific Reports is an impact factor of 4.
Yeah, I feel like it's suited for a smaller journal just because the results aren't anything like groundbreaking. Like, it shows kind of what people would think. Like, your brain deteriorates with drug use. Like, that's what people assume, I think. So it's not anything like super significant.
Ketamine is showing remarkably good.
It shows good potential of like for other uses, but
yeah, but specifically those other uses are post-traumatic stress disorder, intractable pain disorders, I think like fibromyalgia and stuff, and then the big one is depression, which is, I mean, if you look at it, if you can back off of any judgment you have about drug use is bad. Okay, so any parents listening, I'm saying drug use is bad, but if you can consider them as, if you consider drug users who use Special K, as it's called, do they have any mental health disorders that would drive them to self-medicate specifically with this drug? You know, what you find in people with mental health disorders is that they do self-medicate in one way or another. Some might exercise, some might, the most common one is alcohol. People drink away their sorrows, which is a horrible way to deal with it, because alcohol is literally a depressant. But ketamine is showing kind of permanent positive affective changes, which is a kind of the holy grail of work in depression, because you can take an antidepressant which dulls your ability to sense the world and have emotions. Like, a lot of people don't understand that about antidepressants. They think I'll take an antidepressant and I'm going to feel better, I'll feel good. And that's not really the case. You will just feel less, not just less sadness, but also less happy. You're not going to be the dynamic range of your emotions is not going to be as wide ranging. And so you might, if you're interested in ketamine as a therapy for that, you might be interested in reading about what this type of study on antidepressants themselves, what parts of the brain are actually affected, right? And kind of in that vein, if any of you found this interesting, Like I said, scientific reports is about an impact factor of 4. To put that into perspective, that's about the same as a general like field journal, like the Journal of Biological Rhythms for circadian scientists. That's about a 4. So it's good. It's not great, but then you compare it to something like Neuron, which is 14, Nature Neuroscience, which is like 16, Nature itself, the journal Nature, which is the parent journal, is like 36. And that's the big sexy ones are all in the 30s. So Science, Cell, Nature, eLife, that sort of thing. Those are all really, really high. So this is a relatively low impact factor journal. And if you don't know what impact factor is, just really briefly, all it is how much impact do articles in a certain journal have on the field, and it's measured by how many citations they get the previous several years. So in a journal like Nature, everybody's citing those lots and lots of times. Whereas in a journal like this, they're not being cited as often because they're not as elegant. But I was saying, in this vein, I've actually gone into the resources page of the AMI where you found this paper and added a new folder, and I called it Ketamine Reviews. And I am going to start doing this regularly, where I am going to provide extra reading for you that you do not have to do. It is there if you get interested in a subject. Ultimately, for your capstone in the AMI, you'll have to pick a field and write a review paper about it. So when something comes across your plate here in Journal Club that you think is particularly interesting, I would go do that extra reading, because you might be able to then easily come up with a review paper on it. And this is this the first time we've done a drug of abuse in Journal Club? I feel like we haven't really done any. Older people, seniors, Campbell, RC, Marco.
I
I
don't
think
remember.
the first time.
Yeah. You think so too, RC?
Yeah, I don't remember anything else. Like,
Yeah. And isn't that strange? Because I thought when I first started this with you guys, I really thought it'd be like every other week we'd be doing a drug of abuse. Because you all are drug addicts. So anyway, okay. So that's a weird. Here we are saying the conclusion that Brendan said was that it is changing the volume of gray matter in certain areas of your brain, with the caveat we don't know if that is a negative thing or not. That is different than, say, I don't know, going around catching syphilis. Syphilis came up in one of my classes today. Syphilis is a venereal disease that has classic symptoms, one of which is hallucinations because your brain is being turned into Swiss cheese. Another drug that would fall into this category is ecstasy. When I give talks at schools about drugs and they ask about this sort of thing, and they ask like what drugs are safe or not safe, I really think that MDMA, which is ecstasy, is one of the worst ones that you can take, because it causes permanent brain damage the first time you use it. And we do know that brain damage is not good. It actually does have negative effects. Whereas ketamine seems to alter brain connectivity and volume right away, but we're still not sure whether that is a totally negative thing or not, which hence its therapeutic potential. Anybody? I really wish this wasn't me lecturing. I really wish you guys would be talking back about it, like other drugs that might be of interest in this manner. Anyone have like wondering about other drugs, and what drugs? I'm waiting for a sophomore to come in and talk about pregnancy. Sophomores, Veronica. Nothing. You guys are desperate to go see the football game, huh? Okay. Well, this was torture. But thank you to those who actually covered the figures for him. Anything else you want to add, Brendan?
If you want to talk about medical value with drugs, Schedule 2 drugs are supposed to have some sort of medical value. Although I don't see a whole lot with meth and cocaine. That's just me.
Right. Do you know, this is nuts, okay? Do you know that meth is actually a prescription drug? It can be prescribed in the United States.
For what?
Methamphetamine. Crystal meth.
Well, ketamine is used like frequently, but I've never heard about meth being prescribed.
Yeah. Hey, if you look it up, it actually has a brand name that's used. It's used for severe cases of adult ADHD, I believe. But it's really not used. No one prescribes it anymore because it's so addictive, so bad. But yeah, I mean, oxycodone with Percocet, that sort of drug, that's a Schedule 2, has some medical value. Heroin's a Schedule 1. Cocaine, what is cocaine? A Schedule 2 or 1?
The Schedule 2, but I don't
Well,
see for it before.
Cocaine used to be an anesthetic.
Oh, really?
Yeah, it was used for eye surgery and for dental. Yeah.
Well, it was also in soda, so.
True. Coca-Cola. Yeah, for you youngins who didn't know that, yeah. Coca is the cocaine, which was added as a little pick-me-up with the caffeine, make your heart explode. Okay, well, thank you, Brendan. Good job. All right, next week, who's scheduled? I forget.
I think I am.
Alex, yeah. Alex, I might talk to you about maybe skipping a week and having, I might have somebody come in for a tea time. We're just trying to schedule things around this. It's a crazy schedule right now. I'm hearing that the Bishop's Mass this week has been canceled. Though I'm not really sure. I'm pretty sure that it is. Otherwise, yeah, okay. Well, I will keep you updated and post Alex's paper as soon as possible. Do you have something in mind?
No.
Okay. Well, thank you, everyone. Alabama or Georgia?
I want Georgia to win, but
Well, you're
Alabama's probably going to win.
wrong. I can't stand either team, but alas, because of Vanderbilt. If Vanderbilt wasn't in the SEC, it'd be a ranked team. Convince me I'm wrong. All right, see you guys later. Those of you who want to stay with Kate, stay back.
Thank you. Have a great day.
Thank you.
Thank you.
Bye.
I have to leave twice. See ya.
See you later.